Hello, my dear students. Welcome to biology class. In yesterday's class, we shall discuss about some aspects of plant breeding, like how to develop the plant for disease resistance, how to develop the plants for insect pest resistant, or how to develop the plants for increased food quality. Also, we have discussed briefly about the concept biofortification. Right. In today's class, we shall discuss one of the important aspect of food production or enhancing food production, that is the single cell proteins. Okay. So, whenever we say the word protein, it is one of the major biomolecule required for our diet. Okay. To maintain all the cellular reactions, protein must be supplied in our diet. Okay. For that, we have found a way, that is single celled organisms. Single celled organisms are microbes which are grown in large scale for the purpose of proteins. Okay, means to get protein, our food must be rich in protein. So for that, we are growing those single celled microbes, organisms. Those are rich in the proteins. Okay. So here they must be not only rich in protein, they must be rich in protein first. Any organism that we are growing, that organism must be rich in protein. Along with protein, it must be rich in the carbohydrates. Rich in carbohydrates. Along with that, it must be rich in healthy fats, and some of the vitamins and some of the minerals, etc. Okay. So for this purpose, we are using three types of living organisms. One is Spirulina. One more is second one, agaricus mushroom. Third one, we are using the chlorella. Okay. So you have discussed this in first PUC, second chapter, biological classification, all about it. Okay. So here Spirulina is a cyanobacteria. Chlorella is also a cyanobacteria, and mushroom is one of the basidiomycetes fungus. So all these are used or grown for supplement of the protein and many more nutrients or the minerals. Okay, we shall see one by one how they are being used in production of the food. First of all, remember our normal food crops or food grains, or the grains contain comparatively less amount of the protein, very less amount of protein. For example, about 3 to 6 kg of grain means the nutrient present in the 3 to 6 kg of the grain are equal to the nutrient present in just 1 kg of meat. Means just 1 kg of meat contains the equal amount of the nutrients present in 3 to 6 kg of the grain. So only they went for the alternatives like this. Also, you may remember that a cow that is of 2 50 kgs, a normal weight of the cow, okay, that will feed on about 20 to 30 kg fodder per day. 20 to 30 kg of the fodder per day. But what about the protein? It just gives about hardly 200 gram of protein per day. Hence in that milk the protein amount will be just 200 to 300 gram. Okay. But looking at one of the microbe, Methylococcus, this bacteria is about just 250 gram weight of this bacteria, but it yields about more than 250 million tons in its lifetime. Means because of its rapid growth, it gives a large amount of the protein biomass which can be used as a single celled protein. Okay, like that our crop plants, our crop feeds, grains or the cereals or pulses, they have a less yield compared to other sources of the food. So only we went for the single celled protein. Single celled protein simply means the single celled organisms rich in protein. Okay, mainly rich in protein. Along with that, they should be having many more other nutrients like carbohydrate, healthy fats, vitamins, minerals, and some of the nutrients, etc. We shall see those one by one. Okay, so all of you know Spirulina is a cyanobacteria. Cyanobacteria, also blue-green algae. They are also called blue-green algae. Okay, it's a rich source of protein. Because of this reason, Spirulina is most commonly used by space travelers. In the different spacecrafts to different planets wherever they are traveling, because of its rapid growth within a short period of time and rich protein content, it has been used by space travelers. Okay, and it is very easy to grow. It is very easy to grow. Grow on any media which is rich in starch. Okay, grow on any media rich in starch. Starch like paddy straw, like molasses of sugarcane, or any of the crop waste. Okay, or any starch industry waste of starch industries. Okay, can grow Spirulina on any one of the medium, any one of the material, plant, animal, etc. if it is rich in starch. So only it is easy to grow and produces a rich amount of the protein. Hence it has been commonly used by the space travelers for food purpose, or also by the use of protein supplement. Right. That's how Spirulina can be grown, and it has been utilized by many space travelers. Okay. Mushroom, you all know, one of the fungus belonging to the class Basidiomycetes. Basidiomycetes fungi. Okay, it produces the fruiting body, the basidiocarp, and its mycelium will be produced in the soil. Okay, and this fruiting body is very rich in a protein, very rich source of the protein. Hence the mushroom cultivation is nowadays become a great agricultural business or extra agricultural business for the farmers. Not only farmers, you can cultivate mushroom in your home also. Okay, it requires a small space, even if it is a 10 to 10 feet of the space in your home or the backyard, you can cultivate the mushrooms in your home. Okay, plan it and you can start up whenever you have got holidays. Meanwhile, Chlorella is also a cyanobacteria or blue-green algae. It also is having the similar kind of requirements for the growth, also similar kind of the nutrient sources, right? Hence, as an alternative to the food crops, these are being used, mainly Spirulina, mushroom and the Chlorella. Okay, so this is about the single-celled proteins, means unicellular organisms or the single-celled organisms that are having a rich content of the protein. Okay, we shall now discuss about one of the main interesting, important last aspect of this chapter, that is plant tissue culture. Okay, we will also study this concept in biotechnology. Meanwhile, we shall discuss a brief idea about tissue culture in this chapter also. Okay, last concept of this chapter is plant tissue culture. You should be knowing that it is one of the in vitro cultivation method of in vitro growth of plant tissues under sterile conditions. Growth of the plant tissue. Okay, growing plant tissue. What is this word in vitro? Right? There are two words, in vivo and in vitro. Okay, imagine there is a plant in the nature. In the nature only, we are giving all the necessary supplements to the plant, like providing water, providing the protection, fencing, etc. Okay, so if any plant, if any organism is taken care in its original place, or original habitat, then it is called in vivo culture, in vivo growth, in vivo conservation. But if the plant is brought out from its natural habitat and given all the facilities outside its natural habitat, it is called in vitro. Okay, growth of the plant outside its natural habitat, inside the laboratory. Okay, so here in vitro means inside our laboratory, right? So in vivo, in nature, in vitro, out of nature, means inside the laboratory, under human care, under human control. Okay, here in the laboratory, by providing all the necessary conditions. Necessary conditions means, first major condition is the nutrient medium. Which is also called food source. also called food source. That food source must contain carbohydrate, lipid, vitamins, etc. Also, nutrient medium, it must contain many more resources. Better we shall mention them: minerals, sugars, proteins. Along with that, it should mainly contain the growth regulators. It should contain the growth regulators. You may remember this concept from the first year, plant growth and development. You have discussed under the title growth hormones. Growth hormones such as auxin, gibberellin, cytokinin, ethylene, and abscisic acid. Okay, but here in plant tissue culture we mainly use auxin, gibberellin, and cytokinin. All these three. These are less commonly used in plant tissue culture. Okay. Also, along with that, we must provide sterile condition. Means without contamination. Without contamination. Contamination means no bacteria, no fungus, no virus, means no infection from the pathogens, no infection from the harmful organisms to the culture. Okay, usually they are grown in a Petri plate in the laboratory. In the Petri plate, the nutrient medium is added. On the nutrient medium, the plant cells will be grown. They may take a root tip or the shoot tip. They can use meristem, or they can use any of the freshly growing plant material. Usually the growing tip of the plants will be selected for growth in the plant tissue culture. Okay, so this medium should be rich in many more nutrients, also growth hormones, and it must be free from the contamination. Contamination here refers to the pathogens or the contaminating organisms like bacteria, fungus, etc. Okay, that's how these are the basic requirements. Okay, and in plant tissue culture we are using the plant material. The plant material used is called explant. Explant, that is any plant material, plant part that is used to grow a complete plant. Under in vitro conditions. In vitro conditions. And this capacity of the plant material, capacity of the somatic cell is called Toti potency. Okay, remember, it may be a question for 1 mark in your theory examination. Toti potency, that is ability or capacity of somatic cells to develop into a complete plant, complete organism. Here it is a plant. Complete organism or a complete plant is called Toti potency. Okay, this property has been identified by the scientist Haberland and Stewart. They have discovered the property in the somatic cells that they can form a complete plant. ಒಂದು ಸೆಲ್ ತೊಗೊಂಡ್ರೆ ಆ ಒಂದು ಸೆಲ್ಲಿಂದ ಕಂಪ್ಲೀಟ್ ಪ್ಲ್ಯಾಂಟ್ ತಯಾರು ಮಾಡಬಹುದು ಎಲ್ಲಿ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ನಲ್ಲಿ. ಓಕೆ, ನಾವು ಯಾವ ಪ್ಲ್ಯಾಂಟ್ ಸೆಲ್ ತೊಗೊತೀವಿ ಅದಕ್ಕೆ ಏನು ಕರಿತೀವಿ, ಎಕ್ಸ್ಪ್ಲಾಂಟ್ ಅಂತ ಕರಿತೀವಿ. ಆ ಎಕ್ಸ್ಪ್ಲಾಂಟ್ಗೆ ಒಂದು ಕೆಪಾಸಿಟಿ ಅದ. ಏನು ಕೆಪಾಸಿಟಿ? ತಾನು ಅದಕ್ಕೆಲ್ಲ ಸಫಿಷಿಯೆಂಟ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಮೀಡಿಯಮ್ ಪ್ರೊವೈಡ್ ಮಾಡಿದರೆ ಒಂದು ಕಂಪ್ಲೀಟ್ ಪ್ಲ್ಯಾಂಟಾಗಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಆ ಕೆಪಾಸಿಟಿ ಏನು ಕರಿತೀವಿ? ಟೋಟಿ potency ಅಂತ ಕರಿತೀವಿ. ಓಕೆ, so and here we have 3 major techniques used in a plant tissue culture. We shall discuss those 3 techniques of a plant tissue culture and what are the uses and where they have been used. Okay, remember these 2 definitions for your theory examination. One more questions. So we have 3 major techniques in a plant tissue culture. First one, micropropagation. Second technique is meristem culture. Meristem culture. Third technique is a somatic hybridization. Somatic hybridization. Okay, we shall discuss these 3 techniques of plant tissue culture in detail. Okay, first one, micropropagation. Propagation here is growth. Propagation means growing. Okay, we shall see one by one. First, micropropagation. It means that growing a number of plants using a small plant material, that is what we call explant, in a short period of time. Short period of time is called micropropagation. Simply taking a small plant tissue, with that we can develop many plants. Okay, so micropropagation, propagation of many plants using small part. Okay, so only the word micro for the small plant material. Okay, here these plants will be morphologically and genetically similar to the parent. Okay, so the plants produced are morphologically and genetically similar to parents. And hence they are called soma clones. Okay, just because the plants produced are exactly similar to the parental plant, hence they are called soma clones. And the stable variations produced during the production of the soma clones is called soma clonal variation. Soma clonal variation. Okay, the plants produced during the production of many plants using small plant part is called a soma clone, because they look morphologically similar, genetically similar, hence they are called soma clones. And there will be a small soma clonal variation or a small stable variation. That is, a stable variation is called soma clonal variations produced during this production of soma clones. Okay, it has been used to grow some of the crop plants like tomato, banana, etc. Okay, simply it says that take a small part of the plant, using that you can produce many plants. You can produce many plants. That is the main aim of this, right? The second technique, that is meristem culture. Meristem culture. We all know that meristem is the rapidly dividing tissue of the plants. It will be always growing and dividing, right? So this technique is used to grow a plant from meristem. Take a meristem, follow all the techniques of the tissue culture and develop a complete plant, right? So developing a plant using a meristem. Okay, and this technique is used in growing healthy plants from the diseased or infected plants. Diseased or infected plants. Diseased or infected. Okay. Mainly the virus infected plants. Remember, I'll just show you the diagram. There is a plant. There is a plant, right? This plant is completely infected by viruses. Completely infected by viruses, right? But remember, the virus will not infect the meristem, the tip, either the root tip or the shoot tip or any apical or axillary meristem. So this plant part, mainly the meristem part, is not infected by viruses. This plant part will not be infected by viruses, hence it can be used to grow healthy plants. Just because virus may infect the root, virus may infect the stem, leaf, etc., but cannot infect the rapidly dividing tissue, that is the meristematic tissue. Hence it can be used to grow the healthy plants or non-infected plants. Okay, that's how this property of the plants, that is non-infecting meristematic tissue, non-infected meristematic tissues, can be used to develop virus-free plants. Okay, you can expect 2 mark question from this. Okay, so here meristem is taken and grown in a controlled medium of plant tissue culture. And the last technique is the somatic hybridization. Somatic hybridization, which is also known as protoplast fusion. Protoplast fusion. Then what is protoplast? Right. We all know that every plant has a cell wall, every plant has a cell membrane, every plant has a cell membrane, it has a nucleus, it has many organelles, etc. Right. when you remove the cell wall, when you remove the cell wall, so you have removed cell wall from the plant cell. When the cell wall is removed, this complete part is called protoplast. It includes cell membrane, cytoplasm, nucleus, mitochondria, chloroplast, all the parts of the cell except cell wall is called protoplast. Similarly, I will take another cell without cell wall. Again, it is also having the nucleus, nucleolus, all those parts. Okay, this cell is taken from plant A. This cell is taken from plant B, right? Plant A was having some desirable characteristics. Plant B was having some desirable characteristics. The aim is to combine better characteristic of this plant. This character was good, this character was good, height, seed color, fruit color, and here this character was good, this character was good. The aim was to combine the desirable character from both the plants. So, one cell from plant A, one cell from plant B. From both of the cells, the cell wall is removed. Okay, then they are called the protoplast or the somatic parts, somatic cells. Okay, and then they are induced to combine each other. So, it is done by two methods. One is electrofusion or chemofusion. Means the fusion of the cytoplasm of the two can be induced by giving electric supply, electric shock, also by the chemical treatment. Okay, fusion of the two protoplasts by electric current is electrofusion, and the fusion of the two protoplasts by chemical treatment is called chemofusion. Okay, by any one of the methods the two protoplasts are fused. Okay, then this can be further developed under plant tissue culture medium, which is rich in growth hormones. Mainly we need cytokinin for continuing growth, but for the formation of root and shoot we need auxin and gibberellin. Okay, gibberellic acid and many more nutrients present in the growth hormones, right? That is how the somatic hybrids are produced. Okay, since this will be a combination of two cells, it is called a somatic hybrid. And the process is called somatic hybridization. Okay, so combining protoplast of two cells or two different plant cells which are having desirable characteristics is the somatic hybrid, and the technique is called somatic hybridization. Okay, and this is how the plant tissue culture will be done using first micropropagation, second meristem culture and somatic hybridization. Okay, so in this chapter, in this plant breeding chapter, we have discussed about plant breeding techniques like a collection of the plant, selection for the desirable characteristics, then hybridization using the many techniques like emasculation, bagging, cross pollination, rebagging, etc. Also we have discussed about biofortification, means improving the protein quality, vitamin quality, and also we discussed about single celled protein, and lastly we have discussed about the plant tissue culture, which is one of the major trending technique of nowadays agricultural field, because agricultural crop production is must be increased because of the increasing population. Okay, this is all about the chapter strategies for enhancement in food production. Okay, we shall discuss few more things in about plant tissue culture in the next chapter. Okay, so it's all about the chapter. So we are at the end of this chapter, right? See you in the next class with next chapter, that is biotechnology. Till that, keep learning. See you in the next class. Take care. Bye bye.